So basically, the main argument I wanted to make in the talk is that we've had an explosion of data in the myeloma space looking at nucleic acids. This is the blueprint of the cell, the DNA, chromosomes, then going to RNA that defines so-called the transcriptome. Many groups are studying this, but what we argue in my lab is that biology in myeloma and all biology happens at the level of proteins. And so this is the reason we need to study proteins. And what we do in my lab, we use a technology called mass spectrometry-based proteomics. That it's a long name, but really what it allows us to do is look at changes in thousands of proteins in myeloma cells all at the same time. Why are proteins important? Well, these are what are really therapeutic targets, essentially. When we're thinking about new therapies for myeloma, we need to understand what proteins are doing, how their activity is changing, where their localization changes in the cell. What I really talked about in particular is over the past year in terms of an update for 2023 is how my group and several other groups have really tried to explore what are the set of proteins at the surface of myeloma tumor cells. Because we all know about BCMA CAR-Ts, BCMA bispecifics, GPRC5D bispecifics, but there are many other potential targets. Those are proteins at the cell surface. Are there other ones we can use if and when patients relapse on those therapies? We can give them additional options. So what my group is trying to do is find new alternatives. Other groups are trying to do that as well. And I was trying to summarize that work that's been done in the past year, and then also give an outlook about how we can integrate these technologies with newer emerging single-cell proteomic-based technologies. How can we go forward and find even better therapies for myeloma patients?